Yeah, good morning, welcome. I would like to present today the work we do about functional structural plant models at IBG3, and I will also discuss the potential I see within PhenoRob. Some motivational facts that you're probably aware of that we can read, for example, in the FAO reports that by 2015 the world production of food will need to increase by 60%. That's an estimate. And also it's estimated that 80% of this increase will have to come from higher yields, because we have a widespread land degradation and increasing water scarcity, and those limit the potential for even also for yield increase. In addition, we have predicted negative effects that come due to climate change, for example, increasing droughts or floodings. And also, there is lots of discussion about adapting fertilizer management in the view of limited rock phosphate reserves, for example. And so, in, for example, resource-limited environments, most likely we'll have to produce in non-optimal conditions the food for the world population, and so in that case an optimized root system will be beneficial, for example, if there is improved water and nutrient uptake efficiency for different crops. And this can be then important, for example, if you look at trade-based breedings. There can be an interest to breed crop species that are drought resistant, that are very efficient in nutrient acquisition, or also other applications like phytoremediations. You can find examples in the literature. Also, if we discuss sustainable agricultural management, we have to decide about the cultivars we are using and about how is our fertilization and irrigation management. And in this situation, functional structural plant models can help us, first of all, to understand the complex processes that occur in the plant soil systems and on multiple scales, and also to quantify effects of different processes under different soil and climatic conditions. And I would like to start now by telling what do we mean by structure in the structural functional models. And by structure we mean that we simulate with the model the explicit development of the plant in 3D space, and we distinguish between different plant organs. For example, leaf, stem or root organs, and we look then at the topology, the geometry, how they are organized in 3D space, and the morphology, and also at the temporal dynamics of this development of the structure. And then what do we mean by function? By function we mean the description of relevant processes in this structure. And these relevant processes are, for example, soil water flow, root water uptake and transpiration, photosynthesis, carbon flow inside the plant, rhizodeposition and the fate of the rhizodeposits in the soil, also nutrient transport in soil, nutrient uptake by roots, and how all this affects the plant growth. Now most of our work that I will show is on a specific subset of functional structural plant models, which is looking only at the root structure of the root architecture. So now I will focus on this functional structural root models, as I will call them. And so usually we have in this situation a soil which is a three-dimensional domain, which is represented by some numerical grid, and we solve models, partial differential equations that describe, for example, water flow and nutrient transport in the soil with some numerical schemes such as finite volume or finite element methods. And the roots are represented also in this system by connected line segments, and we have a branched 1D grid on which we solve the flow and transport equations. And I would like to illustrate what I mean by a branched grid by first looking at a regular one-dimensional domain, like this line here on the left, which is discretized into 9 nodes. And when we solve a partial differential equation on this grid, whatever, for different numerical schemes, we will at some point end up with a situation in which we solve a linear system of equations represented by this ax equals b. Do I have a pointer? Pointer? No, not sure. Okay. Yeah. And normally we will end up with this kind of tri-diagonal matrix here for the matrix A. And the boundary conditions, that would be the node 1 and 9 here, are represented in this vector B at the first and the last entry of this vector. And if we have a branched one-dimensional structure like this one, then our matrix will no longer be tri-diagonal or diagonal in general, but will depend on the connectivity of the nodes. For example, here 3 and 8 are connected, and so you will have an entry here in the matrix A. And also the boundary conditions are distributed across the vector, and we have more than 2 boundary conditions first of all, and they are at these different, for these different nodes at any position within the vector B. Just to illustrate what I mean by this 1D branched structure, an actual root system looks more like this, so each of these colorful nodes would be one node, and the matrix would of course be much larger than this 9 by 9 matrix that I showed for the illustration. Then when we solve these transport equations, flow equations in both the root and the soil domains, then we have to somehow describe how they interact. So basically we have 2 spatial domains which interact with each other. And we do this by describing it via volumetric sink terms. So here we have the sink term for the root uptake, where we have some flux across the root surface, and we convert it from a flux into a volumetric sink term. And for the soil, we also have to consider a situation in which more than one root segment could be inside one soil control element. Then we have to sum over all the root segments that affect this one soil control element. And these are just examples for what could be this flux term J. So here we have, for example, the water flux, which is dependent on the pressure gradient between the inside the root and the soil water potential at the soil-root interface. The nutrient flux, this is one example, could be described by so-called Michaelis-Menten kinetics, that is dependent on the concentration of a nutrient at the root-soil interface. And so now, if we do this, what can we gain? What information can we gain from such a relatively complex simulation? And this is an example that shows the vector field that describes the water flow in the soil towards the root surface that is induced by the root that takes up water. And we have the information about the 3D position of each of the root segments. We have the information about water and solute, water content and solute concentration in 3D space, and also about the fluxes in the soil, in the root system, and across the membranes. Of course, if we have such a relatively complex model output, it's sometimes not so easy to interpret the results. And so one way to do that is to aggregate the complex information into things that are more easily to interpret, like, for example, 1D depth profiles, like here, where we can have depth profiles of the root surface area density, for example, or the water uptake or the solute uptake. So at which position in the depth profile was the uptake the maximal one? This could be inferred from this information. I just would like to let you also know that we have a benchmarking initiative because with a goal to quantify and compare different functional structural root models with each other. And the goal is to arrive at some point at improved models with which we can simulate various scenarios with greater confidence. Until now, no quantitative evaluation of these models has ever been done. And this is done by a group which was initiated by a workshop by these people you see here. And we disentangle the complex model into 3 different parts. So we have the root architecture development, we have the water flow in soil, and the water flow in roots. And then we look at the coupled systems when we couple water flow and solute in both soil and roots with a static root system. And then when we also include the root architecture dynamics, the growth of the roots. And these are the ones where I will show you examples now. So let's start with only the root architecture. So we want to describe the structure of a root system in soil. And a tool that we develop at IBG3 is a model called cPlantBox, which is a recent extension from a model we call cRootBox. And we simulate root growth processes with this, for example, elongation, branching, death of the root systems, tropisms, so the change of direction which may be affected by, for example, nutrient concentrations. The model is stochastic. That means all roots that are simulated are slightly different. And we use so-called signed distance functions to represent geometries in which the roots has to grow. And with this we can mimic, for example, field conditions or also experimental setups like here in specific containers. It also has an interface which enables us to easily couple this structural model with different other functional models. And it's a relatively fast code programmed, the core programmed in C++, but it has a Python interface which is relatively easy to script. And in addition, we have a shiny Rootbox app which is online available, which enables you to simply online create your own root system using parameters from a database, which is also available. So this cPlantBox models plant structural development, the overall model now, which is the cPlantBox model, which develops the organs, different kinds of organs, so seed, root, and stem. And there is, since it's an object-oriented model, we can easily extend it to further organs. We're thinking about fruits, for example, which are not yet implemented. But it's also possible to just simulate one particular organ type, for example, only the root architecture. And then this model can be coupled to various functional models. And there are examples where we coupled it to DUMUX for water flow and soil transport in the soil root system. We also coupled it to Simplace, a crop model platform, where 1D depth profiles of root length density are computed and passed to the crop model in Simplace. And it's coupled to a model called Piaf Münch for water flow and solute flow in the coupled system of xylem and phloem in the whole plant. Here is a link from which you can download the code. It's freely available and open source. Here is an example of a simulation in which the simulated root architecture is dependent on the nutrient concentration. So that would be a case of chemotropism. And it's an example where we have a pot. On the right-hand side, we have more nutrient concentrations than in the left, in the blue side. And in this top panel here is chemotropism enabled. And so the plant developed many more roots in the pot where there are more nutrients. Whereas here we have no chemotropism and only gravitropism that drives the root growth downwards. We can also simulate multiple roots. Like here, this is an example of kind of a field simulation, which also here these blue tubes should represent the rhizotubes we have at the Seelhausen rhizotron facility. And the roots will grow around these tubes. And then we can use this kind of model to perform what we call a virtual field sampling of root systems. So we can have our many, like 200 plants, root systems next to each other, and then we can crop the simulation result to what we would measure if we now did in this field a soil coring, or if we now did in this field a trenching, where we count the root intersections, or if we do rhizotron, mini rhizotron camera observations of the root systems that will give us root arrival curves at different depths. And so the goal of this exercise is that if we do this virtually with a known root architecture, we can then learn, can we, if we only have this information, which we would measure in the field, can we infer the root architectural traits that are behind each of these plants? So it could be the basis for an inverse estimation of root architectural parameters. Another way to estimate the root architectural parameters needed for the root architecture model, and these are parameters we actually need in a model, things like the basal zone length, the apical zone length, the interbranch distance, the root radius, the branching angle, and so on. And one way is to use 2D images, which are quite abundantly available from rhizotron images, rhizobox experiments, from filter paper experiments. And we have software, maybe I can start, yeah, which automatically finds the roots in an image and then gives us the coordinates of each of these root segments as a result, from which we can then compute the root architectural parameters. So this is an example of a neutron radiography image, which was imaged at 3 time points. That's what you saw in these 3 jumps. Also, we can use 3-dimensional images of root systems. There are several methods that are used, like magnetic resonance imaging, microcomputed tomography, or neutron radiography. And we do have a possibility to manually trace the roots from these images in a virtual reality system we have at the Jülich Supercomputing Center. That means you sit here and you basically draw the roots that you see manually from the image. In addition, there is software developed at IBG2 called NM Routing, which also works well with some of our root systems, and we have good collaboration with Daniel Pflugfeldner there. Also, there is an ongoing collaboration between Jülich and University of Bonn with the group of Sven Binke, with the goal to develop an automated segmentation and tracing, which would improve basically the NM Routing software. Here is also a virtual reality headset system, which is also developed in the Jülich Supercomputing Center, where you can see the root here growing in a kind of a spaceship room. And then we can also do a manual tracing there. The result of such a reconstruction of the actual root systems from a 3D image can either be used directly as an input for a functional structural root model, or it can be also used to compute the model parameters needed for a root architectural model. And so what I would like you to remember from the structural part is that we can reconstruct root systems from 3-dimensional images, and they can be a direct source for the root grid needed in a functional structural root model. Alternatively, root grids can be created with a root system architecture model. For that we need parameters, and the parameters can be either obtained by inverse estimation, then in that case, for example, also field data, or from image analysis using both 2D or 3D images. In that case, they are normally younger root systems. Then we can mimic actually the root growth in a functional structural root model simulation. If we know the root segment age, we can mimic the root growth from a final root grid, but that would mean that the root growth is not affected by dynamically changing soil conditions in the simulation. And the most complex way of making a simulation is to run the growth model simultaneously with a soil model where the dynamically changing soil conditions may also affect the root growth, the root architecture, like in the example I showed you with the chemotropism. And now I would like to come to some examples regarding the functional structural root models, looking at in the coupled root soil system. Just to remember that it's actually a multi-scale problem where we can have the viewpoint of a plant in the context of the whole plant in the context of the soil-plant atmosphere continuum. We can have the viewpoint of a single root with small-scale gradients potentially around the root, and even smaller to the even pore size or particle scale. And I would like to show you now two examples. This is a recent example where we developed a model on solute movement in this soil root system. And what you should know is that we have, again, the soil subproblem where we solve the solute transport equation. We have this root subproblem where we also solve the solute transport equation. And we have a way to couple these two domains via our volumetric source terms. And here you might recognize again the Michaelis-Menten kinetics for nutrient uptake. Now, if you look at this equation, we need the concentration at the root soil interface. And how can we compute this value in such a soil model which has a coarse soil grid, typically in the order of 1 centimeter side length? So we would take, for example, depending on the numerical scheme, these 8 nodes of this soil control element and compute an average value, which we would then put here in this uptake function. But we know that the rhizosphere gradients could be even in the order of millimeters or even smaller. And so one way to handle this could be a brute force higher resolution, to really go down to like sub-millimeter scale. But you can imagine that then your computing time will explode, even if you do local grid refinement. And so what we did is we solved now for each root segment in a soil domain, we solve a one-dimensional radially symmetric rhizosphere model at high resolution. And it's much faster because it's only 1D. It makes the assumption, of course, that there is radial symmetry. So this is an error that we make, but the error is relatively small, as we could show in the paper behind this simulation. And so we applied this model in a recent collaboration with the University of Leuven, where they had a upland rice species growing in different treatments regarding the phosphorus application and the water treatments. So they had 3 P levels and the drying and the field capacity scenario. And then we mimicked, we also had root architecture or root data available from which we could reconstruct the root architectural parameters and modeled the root architecture in the container, and we mimicked, so to say, the experimental setup with the same water and P treatments. And then what could we learn from it? So the one result that was also seen experimentally and that could be reproduced with the model is that in the cases where P was added in different levels, when there was less, in absolute amounts, less water uptake, there was also less P uptake. This was not the case for the lowest P scenario. Then we had the same P uptake even if there was less water uptake. And what additionally can we learn from such a simulation, which might be harder to know from the experiment itself, is, for example, we can tell at which position was the uptake taking place and which roots contributed most to the overall uptake. And in this scenario, the P acquisition shifted from the top to the subsoil under drying conditions. And we could distinguish which root types of this rice contributed more in the drying versus the field capacity case. And the second example I would like to show you is a simulation we did regarding maize root traits on water stress tolerance. And I show this because this is an example how we use the model mimicking field conditions. So first, let's discuss the setup. So we have 3 different maize phenotypes. There is a standard one, a steep, deep, and cheap phenotype according to Jonathan Lynch, and we have an intermediate phenotype. So steep, deep, and cheap is supposed to be a root system which is less prone for drought stress. So it can handle drought conditions better. And we put these 3 plants in 2 different hydrological regimes. We had one with a dry topsoil and a wet subsoil, and the other way around. And then we use periodic boundary conditions on the side to mimic one plant in a field situation with a neighboring plant that has the same architecture. And each plant periodic cell for each plant is that the size of it is determined by the inter and the intra-crop distance. And so what can we learn from this? So we simulated the water dynamics in the root zone for 1 week. And so a 3D visualization looks like this. And now what I would like you to see, so now then we can compute, for example, the black curves are the potential transpiration which we impose as the boundary condition at the root collar. And these colored lines here are the actual transpiration. So that means if it's less than the potential one, all of the plants had stress in the scenario. And then we also plot here the cumulative water uptake over time. And I just want you to see that in this scenario, the blue and the green were the best one, whereas the red one was slightly worse in terms of the amount of water which could be extracted under the same boundary conditions. And here we have the green and the red one. That's the top moist case, and the blue one was much worse. And so you could say, okay, that's good. I just plant the green, that's the intermediate phenotype, and I plant it no matter what my hydrological regime was, it will perform good enough. And now then what we can also do is we can compute, so you can regard as the cumulative transpiration something like a time, and then we can compute what we call the equivalent soil water potential. That's the average soil water potential experienced by the root. So only choosing those soil voxels in which there are actually root segments. And the more negative, the more stressed the more stressed the plant is. Yeah? And so we see that here in this, for example, we have the red one is the most stressed one, but blue and green performed similar, but the green one already feels more, experiences more stress than the blue one. So if we would have extended the simulation time for longer, the intermediate phenotype would have had a worse performance. And the same is in this case where we have the red and the green were the best one, but the green one already experiences more stress. So these are just examples that I wanted to show you what can be applications and what can we learn from such a model. And this is now a first example of an ongoing work where we use the whole plant functional structural model. And here we coupled the sea-plant box model with a model called Piaf Münch, which mechanistically models the xylem and the phloem flow. And we put it in a heterogeneous soil. So one root is in a wet soil and one root here is in a dry soil. And what we learned from it, what the simulation tells us, is that of course we have more water flow from the regions, from the wet soil regions, and then from the dry soil regions. And on vice versa, we have more carbon flow towards the dry soil regions, more carbon flow inside the root, that means. So of course I couldn't show you a complete list of everything we have done. So if you're interested in more examples, you are welcome to contact me, or these are examples looking at exudation, mycorrhiza, root hairs, and also using it in different mimics of experimental setups. This is my final slide, which is kind of what is my view of the role of functional structural plant models in PhenoRob. And here on the right bottom, we have kind of a vision that within PhenoRob, at some point, we want to have at least near real-time management decisions. So I'm imagining a robot or a tractor that drives over the field and wants to be supported somehow in making a decision. And likely the algorithms that will be developed here could be either based on machine learning algorithms that have been developed based on some experimental data training sets, or also using crop models that are also developed within PhenoRob. I don't think that this error could exist. So I don't think the functional structural plant models will have a direct input on a management decision of somebody on a tractor, because the computation time is much too high. Yeah, so I don't think that. But we have functional structural plant models that can help to improve crop models by, for example, upscaling, computing effective parameters, and we have working examples for this, and Jan will show them in the next talk. And another idea, which we haven't had any examples yet, could be that we could use such models to constrain also perhaps the machine learning results. Because here we have some mechanistic knowledge about a system, and maybe that could be used to constrain any machine learning algorithms. And yeah, with that, I would like to thank you for your attention and happy for questions.
Yeah, thank you very much. Interesting talk, at least for me. Open for questions. Thomas, of course. Yeah.
Maybe on your last point,
yeah.
It's something that we're also thinking about doing in project 6.
Okay.
Is because we have a rather computationally expensive economic model of farms, and which would take too long to solve on the fly. So we're thinking about maybe using some of the outcomes of that model and see whether we can generate data with the model that the machine learning algorithm can then use to. So there's actually a PhD student
okay.
started in January, I think, with Jürgen Dahl, who will try to do that with the economic model. So maybe it's something that we could explore jointly, that we have just a different model.
But the strategy could be similar.
Exactly.
Yeah. Thank you. Yeah, that sounds interesting. So you actually create virtual training data sets for the machine learning algorithm. Yeah. Yeah. Yeah, this we could think about in the same lines. Yeah. Thank you.
So a root is not a root. Even though I work with photosynthesis, I know that there are different classes of roots,
yes.
fine roots, coarser roots, which have greatly different functional
yes.
properties and importance also for water uptake, nutrient uptake. Can you comment a little bit how these pretty complex different functional classes are handled in the models?
Of course. Yeah, so I showed you these connected line segments quite early in my talk. But if you remember them, or maybe I should quickly go back. So this is basically how we, in a model, envision the root. And each of these segments here can have attributes. And these attributes can be the root radius, can be the root segment H, or uptake properties that are dependent on the root H, for example. So this would be handled via the properties, the attributes that each of these segments have. And so and what we also have is examples where we have root segment H and root type dependent root hydraulic properties. And that makes the simulation a bit more complex, of course. But the most challenging step is the parameterization of this, because there is not so many data out there that will tell us how does the root hydraulic property depend on the root age. But it's handled technically as attributes on these different root segments.
And this is then handled numerically in the model?
Exactly, because then if we have this functional term here, then this Kr does not necessarily, for example, have to be a constant. So this is the root radial conductance, and it will tell us, depending on the pressure gradient, how easy or not can the water flow actually into the root. And this could be dependent on the root segment age, or on the root radius, or on other properties. And so for each little root segment in the simulation, then this J would be different depending on its attributes. Welcome.
This goes a little bit in the same direction. So basically, if I understand correctly, you're trying to deduce from root morphology and structure nutrient influx also. And we know that, for example, I mean, like challenges that plants face in real life, basically pathogens, insects, and so on, they have a huge effect on nutrient acquisition, and that doesn't change immediately like root morphology. And that is not true. That's also not the same for all nutrients. It's very different for different nutrients, like phosphate and potassium, for example, is very different in this case. So how do you implement these kind of other parameters that cannot be explained by root morphology in order to then simulate or calculate nutrient uptake?
Yeah, so this goes into similar lines like I said in my previous answer. Of course, I didn't show you, and that's because we don't have any examples of simulations including pathogens. Yeah, so this is kind of more like a research gap for these kind of models. What we can do and what we aim for in other projects is, for example, we can also make these properties that are effectively the functional efficiency for root water uptake, for nutrient uptake, and so on. We can make this dependent on any property, on any attribute. So we not only the age or the root radius, but we could make it dependent on, if we had a model that gave us the pathogen, I don't know, a population density or something in a root, we could make all the functions dependent on that. Yeah, but the problem is we don't have, as of now, such a model that gives us the pathogen effects on the whole root system scale for each of the root segments. But so how we would do it if we had this information would be, again, to make these values here, which describe our function, as a function of pathogen density, for example.
I just want to clarify what you mean by dynamic and stochasticity, and basically understand whether essentially everything, except of course for the Vmax, is a linear function.
Definitely not linear. So I didn't even go into much of the details. I just showed you water flow and solute transport processes. Water flow is an example of nonlinear model because it's highly nonlinear. The hydraulic conductivity is highly nonlinear with water pressure in the soil. Or what you mean by nonlinear?
So
or you mean the growth is linear?
All
or?
of it. Basically, the fact that the rates over time can change with both root development, root function, and responses with the shoot. So I was wondering to what extent the model is capturing changes in rate of dynamics, and what you mean by the word dynamics, and what you mean by stochasticity.
Okay, so then I focus on the root model itself, yeah, in my answer. So the root growth doesn't have to be linear. So each root elongates according to a growth function, and the most commonly used one is linear growth. That's very often used, that we say it's growing constant with a constant rate over time. But you also have lots of examples in which the root growth is nonlinear. For example, decreasing with as the root approaches its maximal length. So then we would have a nonlinear root growth. we can also, in our model, you can specify as a user any function you want, as long as you can parameterize it, and give it to pass it to the simulation so that the root growth can be according to any function. But the most typical ones are actually either linear or non-linear, in a way that the root slows down its growth rate as it approaches its maximal length. So this is what I mean by the root growth. And then the stochasticity is not directly related now to the linear or non-linearity of the root growth.
No, but this allows you to kind of put a dynamics of functionality into the overall system, right?
So what we do is for the root architecture development, you have seen the list of parameters. Each of these parameters has a mean and a standard deviation, so is described by a probability density function. And then in a specific realization of a root system, each root has a different set of actual realizations of these parameters coming from the same probability density functions. So in this way, then each root will be a little bit different.
Okay.
One will be a little bit longer or shorter, and so on.
Okay, I got it. Yeah. Great.
Okay, perhaps you can show us the previous slide number 7.
This one or
Yes,
this one?
Yeah, it doesn't matter. I'm referring to the equation A times X equals
Yeah.
to B. It was not clear to me where comes B from. I mean, we have these type of equations very, very often in geodesy, and in our case, B is usually an observation.
Ah. No.
And what is you called it boundary condition or
Yeah,
whatever.
yeah. No,
where
that's
comes this data from? You
this.
need some
This is not at all necessarily, there is no data. So not necessarily there is any single data point in this.
Okay.
So this comes from the discretization of a partial differential equation. So because we solve here a partial differential equation on some domain, right?
Yeah.
And if you solve it numerically, then you have to discretize in time and you have to discretize in space.
Sure.
And depending on the numerical scheme, whether you have an explicit or an implicit numerical scheme with time, and depending on whether the partial differential equation is nonlinear or linear, you have to also have some linearization step. But at some point, through this discretization step, you reach at this system of algebraic equations which needs to be solved. So this is a result of the discretization procedure of the numerical scheme for solving the partial differential equation. Okay. Yeah, and typically here you have in this vector B basically this is what you could call the data, in the sense that you need to solve a numerical model with some boundary conditions.
Okay.
And so here are the boundary conditions typically in such a scheme.
Okay, but nevertheless, somewhere data has to come in.
Yes.
Especially when you look on your last
Of course.
slide,
Yeah.
or the second last one, in the field or whatever, something is happening.
Yes.
Of course, this is simulation, I understood, but of course, somewhere you have to introduce some data.
Yeah. So
what kind of data is it and where?
Okay, so I showed you that the data we need, so the model parameters, let's say, we need. So for the root architecture, yeah? If we look at that again.
Yeah.
So so this is a list of model parameters. We need to run the model.
Yeah.
And these values here, they have to come from some kind of data, right? And so I try to show you that we have data that are basically images of root systems, like 2D images like this one, or 3D images, and then we can process and analyze these images so we can infer these data, these values. And then the second thing is, once we have a model result, so we have like a 3D explicit architecture, we can compute aggregated results like root length density profiles, what we would measure in the field. then we could also infer the root architecture parameters through inverse estimation by comparing the model output with measurements. These are things we can measure in the field.
Okay.
So,
I guess, yeah, I mean, so that does roughly look like a soil core profile that comes from the field, except we get much more variation in that shape, in reality, with real data. So like this, or like this, or like this, or...
yeah, this
so
is just...
validate
Yeah.
this now with real soil coring?
Well, this is only a virtual. So what we actually did is we did a literature review on what do these profiles look like, and we compared model outputs in general, whether it's a realistic shape or not. And the next step is that we actually, that's the next step. So we haven't actually used this method on real data. But that's actually a next step that we are interested in doing.
A question related to that one. So you could actually take the entire simulated field and make a root distribution and then see which method, like soil cores, trenches, or level tubes, would actually the best to catch the
that's the idea.
So what would you say is the best one? Which method?
I don't have an easy answer, sorry, at this moment. Because, so what it can also help you is to design your sampling scheme, for example. Yeah, we can say, okay, we need 10 soil cores or maybe 50 soil cores. We can tell what's the variation we get, and we can see if we have so and so many data, and does it does it then represent does the model represent the field data, or vice versa, to help you to design such a sampling scheme? Yeah,
but
that's it.
Immediately, a trench will take you probably 2 weeks
Yeah,
to analyze.
I know.
Soil cores are much quicker.
I know.
So they all.
Yeah.
So in that sense, it is very powerful to do a complete, let's say, value proposition on the different methods, given the time, the input, the land, all the factors, because these are 3 very, very different.
Yeah, so this is something where we developed the method now, but it's not in a stage that we have applied it to a lot of data. So I don't have like the answer, which is the best sampling scheme. But I would also say it depends on what you want to do with the data. But it could also, so there could be a different answer for a different aim of what you want to do. So.
Yeah, thanks, Andrea, for this interesting talk. I have a question. I mean, you described quite succinctly the added value of these functional structural flag models for realizing the PhenoRob vision. I would be interested in learning, what do you see as the key challenge right now from the PhenoRob perspective in your research, and where do you see progress in the next 1 to 3 or 5 years? Like, if you had to put it on a timeline, what would be the sort of the big steps that you now need to take in order to contribute to realizing this PhenoRob vision?
Okay, so that's a very good question. Maybe I can go back to my last slide. Yeah.
Yeah.
Oh, sorry. So here. So for me, so this one is far from being realized, right? I don't know actually so this one is an algorithm which is, I guess, starting to be developed, yeah? So I think, and I am personally not aware how far this process is, to be honest. So I think one challenge would be to have a communication and like sort of to have a communication between the people who do this and the people who contribute directly or indirectly, like us, to this development. So for me, that's the first challenge, yeah, in terms of communication. The second thing is here we are quite sure for at least root water uptake what we want to do. Here there is a scientific challenge to extend this to other processes like nutrient uptake, for example. Or the biggest challenge is actually to put the biology in terms of microbiome in there in this kind of models. Yeah, so that's a big challenge scientifically, and that could be very interesting for PhenoRob. And then here, this path, there we have a PhD student starting soon who in CP2, which will sit in between these two topics. So he will work on functional structural plant models part, but he will also at least be a user of the machine learning part. But also here, I think there is a lot of communication that's still needed. So there's on the one hand, what do we want to do with who, and to do it on a good timeline together. So for me, that communication is a key. And then for the science, and then also here to develop the machine, maybe even to create virtual training data sets, whether that's of interest for the people who do the machine learning for this kind of models. Here we have still scientific challenges in terms of the development of the functional structural root models. And the keyword for me is microbiome there.
I like Laura's last question. I have one question just with respect to the phosphate experiment. So you showed that drought has a stronger impact on phosphate uptake in soil that was supplemented with phosphate as compared to
yes.
that was not supplemented.
Yes.
And it seems actually a little bit trivial for me, imagining that maybe under a supplemented condition there is some part of phosphate is coming to the plant by bioflow, whereas in a low nutrient immobilized P condition, you need immobilization processes, maybe acidification, carboxylate extrusion, and so. What's your explanation of this, and how did you supplement phosphate in the soil? It's not, how would you interpret your results, basically? What's your explanation?
Yeah, so that's a good question because so the experiment wasn't done by us, yeah. But the experiment was done in a way that there was a low P soil, so the subsoil P was low in all the cases, and then the topsoil was either supplemented with P at these two different levels or not. So, I think just phosphate, yeah. But this we can look it up together if you're interested, yeah. And so we have one situation in which there's really a very suboptimal situation for P, and the one that's called sub-P does have at least some P, so it's not really starving for P, and then a plus P is one that's really optimal. So that's how the setup was. And for me, it was interesting to see that in the one case, actually the water flow or the water uptake affects the P uptake, and in one case it doesn't. But yeah, for us, I don't know if you say it's trivial, but for me that was interesting actually to try, and we are also still analyzing a bit why, because what we are doing is we're screening which are the roots that contribute to the uptake. In which depth is the uptake occurring? Does it coincide with where the water is or not? And so on. So,
what could be going on there, I'm not sure, is you could be generating an osmotic stress in a high P condition, particularly up at the boundary of the rhizosphere. This is one thing to look at, like some of the work of John Pasira's. The other thing is in the high P scenario, you might have had further to that water demand situation, greater transpiration as well.
Definitely.
So both at the, that could be possibilities. Be interesting to see.
So the transpiration was definitely different in the different cases.
Then you have a higher salt condition. When the nutrients are high, at the root soil interface, this was some older work that John Pasira did.
Yeah, well, that depends on the concentration. Yeah, we see this, and I think also Jan will show an example where we have salt accumulation near the root surface. But this is for sodium, where we saw it, and we had some data also to show it. For phosphorus, I'm not sure if that's even at the high P treatment occurring. So
it's a problem with N.
Yeah, for N I can see it, because of course N is not very strongly buffered in the soil, so you would have quite a high convective transport towards the root surface, and then you will have accumulation. Yeah.
Okay, so one final question, more small remark from my side. You argue that this is the final goal of PhenoRob real-time management decision. It could also be a breeding decision.
Of course, true. So
the timescale is a little bit different, of course. But
yeah. So I tried to mention it in the beginning but here, so it's missing. You're right.
So, many thanks.
Welcome.